So just to wrap this up, the title of this symposium was leveraging or address leveraging gamma delta T cells and T cell biology for cancer immunotherapy. And I think there's, we've heard from the talks and from the discussion too that there are various features of gamma delta T cell biology that could define an upside potential, could define a real advancement in clinical use as opposed to approaches we currently have. And clearly, we saw that there's data that's now being confirmed that their presence in cancer tissue is actually correlated with improved prognosis and survival, and that's not just in one cancer type, but it's actually across multiple cancer types. We've seen data that says that they have tremendous cytotoxic potential, and I was very intrigued by looking at the serial killing experiments that were done in one of I think it was in Michael's talk that showed and that clearly show great potential. What's important that everyone recognized is that their killing is independent of MHC class 1, class 2 downregulation, at least in most cases, although there may be some exceptions. And this downregulatory mechanism is very, very common in epithelial and solid cancer. So I think this is one way to get around that potential immune evasion mechanism. And we know that, at least as far as we understand it, the cytotoxicity of these cells is triggered by target cells that are in stress. That includes malignant cells as well as infected cells. And that stress signal, or the stress patterns, if you will, that are expressed either through butyrophilins or other targets, are less conspicuous on healthy tissue, and therefore we may expect to see minimal cytotoxicity towards healthy tissue, and perhaps even a greater safety efficacy window when used in the clinic. So what we see in terms of translation of this biology now is sort of 2 or 3 different approaches, but they're converging on the, let's say, the enthusiasm for this approach. One is clearly focused on engineered cell therapy, starting with allogeneic V delta 1 positive cells, which the development of that platform is clearly ongoing. And here you can ask the question: how does introduction of a CAR help or enhance the applicability of these cells? What about delta 2s? I think we also saw clear evidence that we could transfer gamma delta T cell receptors and their properties onto engineered alpha beta effector T cells, and then get improved cell therapy, at least improved in principle in all the experiments we've seen, and hopefully this also translates into the clinic. Very intriguing, working on butyrophilin and butyrophilin antibodies, as exemplified by the BTN3A antibody ICT01. Clinical studies ongoing. I guess those expansion cohorts expected, ongoing studies in solids as well as liquid tumors. I think this is intriguing biology, and mode of action of these antibodies, as it gets better understood, may actually guide us into very effective clinical development in the near future. And then the last platform we heard during the presentation, but that also seems to be underpinned by a lot of preclinical data and understanding of mode of action of these engagers, is actually targeting or using V gamma 9, V delta 2 specific gamma delta T cells, and engaging them in a killing activity towards a target of choice that could be encoded by the other arm in a bispecific or even a multispecific engager. We know there are studies ongoing in multiple myeloma. Other studies are being started in prostate cancer. And the question here really is, does this provide us leverage to broaden the target space we can use for targeting cancers, or does this actually allow us to go back to well-described targets that did not seem amenable to the use with CARs or T-cell engagers, but now with the potentially enhanced safety efficacy window, can we get there? So the clinical data will start answering very important questions about safety, obviously, about cytotoxic potential and durability of that cytotoxic potential. clinical activity as it translates to durability of clinical response, and can we show this beyond hematological cancers? We addressed this, I think, well enough. There's clear potential for combination therapy, and as Pam reminded us, the future is probably not single agent application. The future of cancer therapy, expanding the benefit of immunotherapy of cancer to as many patients as possible, is very likely dependent on combinations and well-chosen combination. And then finally, a question that we're not going to address here or in these specific oncology studies, but there is a potential to utilize gamma delta biology beyond oncology. So what's exciting, and I think we heard the excitement today, is that there are programs that are currently listed as in phase 1 or beyond, or even very close to that. And these are going to deliver clinical data, but hopefully also a lot of biomarker or pharmacology data that allows us to understand and more rationally design gamma delta T cell therapies based on either platform. From the cell therapies, we've seen evidence from, or we're going to see evidence for the allogeneic application of unmodified and genetically engineered V delta 1 T cells in various indications. We're also going to see gamma delta TCR engineered alpha beta T cells applied to multiple myeloma and ovarian cancer. At least that's what's listed, and I'm sure there are multiple other plans. From the T cell engager side, we've seen data that led to starting a trial in multiple myeloma targeting CD1D, and we're going to see hopefully data, not just clinical data, but also immune monitoring, immunopharmacology data targeting PSMA in prostate cancer and beyond. And then finally, I think there's very intriguing data coming out of the butyrophilin targeting antibody programs, and we haven't gotten to the bottom of that, but it's really very promising to see early clinical signs and signals of potential activity, and clearly these can be applied to patients in a manner that looks like it's very safe. So without dwelling on this too much, I think I'd like to close this symposium and this session. Thanks to all the speakers. Thanks to Daniel, Jürgen, Michael, Hans, and a very special thanks to Jim and Pam for joining us for this session, and especially for the panel discussion. Thank you all for joining, and I would say thank you to the Antibody Society for helping us to get this program up and running. Have a great day.